So we'll just quickly go back through what we talked about here. We went through classification or features of the animal kingdom. So just to quickly go back to the list, eukaryotes, right? That's a given. All animals are eukaryotes because every group we're talking about that isn't prokaryotes, so not bacteria or archaea, is eukaryotics. That's pretty straightforward. All animals are multicellular, no exceptions. Specialized tissues in just about all groups except for one group. Do you guys remember which group that was that doesn't have specialized tissues? Okay, we're gonna go over it again. Yeah, the parazoans, right? And that's your sponges. So yeah, let's talk about all the animal groups as we go through 28, 29. We'll touch on that some more. Motility, what does that mean? Animals can do what? Yeah, they can move on their own power, at least at some point in their life. So you may see animals that are sessile, that are rooted down, but that at one point or another they can move. So maybe they're larval phase. Heterotrophs, what does that mean? What does a heterotroph do? Yeah, they got to get their nutrition from other sources. They can't photosynthesize, so no animals can do that. There are animals that have symbiotic relationships with photosynthesizers, and they can kind of co-opt that activity. So we'll look at some examples of that. But on their own, there are no photosynthetic animals, only heterotrophs. Most reproduce sexually. We took a look at that. There are exceptions. Life cycle is diplontic, meaning that you're diploid except for your gametes. So there's a single-celled haploid stage, which is different from plants, right, with alternation of generations. And then follow a plan, a predetermined body plan that's programmed in your DNA. So those things are just characteristics of the group. We looked a little bit more at, there we go, tissues. These are the 4 types. I told you guys just to be familiar with function and examples of those. We looked at the tree a little bit. Here's where we introduced parazoans, the sponges that are over here on this branch, sister to all other animals, but a basal lineage. Are you guys comfortable with that terminology? We'll talk about that a little bit more again in the next chapter too. And then we looked at reproduction. We talked about metamorphosis. Everybody knows caterpillars turn into butterflies and tadpoles turn into frogs. And then some animals just come out like small versions of the big thing, like we do, right? We talked about a couple of examples of asexual reproduction, so just be familiar with the definitions of those. Embryonic development, we spent a very short period of time talking about this really complex process, okay? But you guys don't need to know any of the major complexity of embryonic development except for the stages that are listed here. So I do want you to know what a zygote is. What's the zygote? How many cells is it? Single cell, right? Is it diploid or haploid? Diploid, yeah. So where does it come from? What has to happen to give rise to this? Fertilization, yeah. So gametes fuse, right? Fuse cytoplasm first, fuse nuclei, first cell with a diploid nucleus, that's your zygote. Mitosis comes next, right? Or cleavage. The 8-cell stage we said was notable because differentiation begins here. Right? What's differentiation? What is the cell doing when it starts to differentiate? You're starting to, yeah, you're heading that way, right? You're getting towards a new function. So you're starting to become something other than just an identical cell to all the rest of the cell. Starting to take on later specialized roles, so to speak. Not yet, but getting there. So the first time you start to see cells even becoming a little bit different from one another is at that 8-cell stage. Okay? Moving towards specialization into those germline tissues that we talked about. Blastula is important because you've got that round hollow ball of cells. It's not empty, remember it's full of fluid, but it's a hollow ball of cells. It's notable because it's going to undergo gastrulation. Okay, remember gastrulation leads to a gastrula. So I want you to know blastula, and I want you to know gastrula. Okay, the gastrula is this stage where you have that infolding of the blastophore, right? And a pore is what? Anytime you hear the word pore, what do you think? Yeah, an opening, some sort of a hole or opening into something. So your blastophore forms as that tissue starts to fold in. Well, it's not tissue yet. Let's say layer of cells starts to fold in on itself. And what is this opening going to become? You guys remember what that tube is going to turn into? It's not spinal cord, not yet. That's later. Yeah, it's going to be the digestive tract. So this is either going to be your mouth or your anus, depending upon your classification. And we'll look at that as a classification feature, whether your digestive system forms front to back or back to front. Okay, so that's something that we're going to talk about here shortly. So the gastrula is important because that begins that first formation of that inner tube that will become your digestive tract. But it's also important because these infoldings are going to become endoderm, and the outer layer will become ectoderm. And in more complex animals, you'll get mesoderm forming in the middle. So the gastrula is that first stage where you begin to see what will eventually become those germ-line tissues, those germ layers. Okay, that are going to then further specialize into the rest of the cells. And the whole idea behind even talking about this, there are two things that are important. First of all, this pathway is shared across animals. Okay, so there's homology here. Okay, so there's a shared ancestor that did this. Okay, that gave rise to all the rest of the animals that do the same thing in largely the same steps. Okay, same fashion. The other thing to sort of keep in mind is that without these specialization steps, without these differentiations of one cell to the next type, getting more and more specific, you are one giant blob of trillions of cells that all look like a zygote, right? That wouldn't be a functional organism. So you're complex because you have specialized tissues, because you have cells that do specific jobs. An eyeball cell is very different from a skin cell, which is very different from a cell that lines your intestines, right? So that specialization is the process. You start out with just the zygote, which gives rise to everything else. But along the way, at each division, at each step, past at least the 8 cell stage, something is changing. Genetic pathways are being turned on and turned off. Gene expression is changing, upregulating, downregulating, so that you end up with your end result of these cells with a very, very specific function. So those are the two things to keep in mind. Cellular differentiation leading to specialization, yes, and the shared homology across animals. Okay, so that's the big themes I want you guys to get across in embryonic development, but I also want to make sure you're comfortable with the terms, okay? So know those bold terms. All right, so that gets us up to speed. Questions, comments, concerns before we go forward? You guys ready to talk about Hox genes? This is one of my favorite things. As you guys know, I like all the things, so you're probably not feeling surprised to hear that. Hox genes are really cool regulatory genes that are found in all animals. They were discovered in the late 20th century, so it's a relatively new discovery. So there's still a ton of research being done to figure out functions and roles and relationships and homology, and it's all very exciting. Okay, all really cool research stuff going on. Hox genes are a class of genes, a big group. There's a whole bunch of different Hox genes. Hox is short for homeobox, and it has to do with just the regions of the genes that turn things on and turn things off. So they're responsible for determining body plan. And we're going to watch this video here in a second, so I'm not going to go into a tremendous amount of detail until after we watch the video together, and then we'll discuss. But what's basically happening is that these are master control genes that are turning on really early in that development process, in embryonic development. And those master control Hox genes are turning other genes on and off downstream. Okay, so the interesting thing about these is that without those master control genes telling you when and where to turn things on and off, you lose the ability to have a predetermined body plan. Okay, so Hox genes are going to tell you things like, how many body segments does this organism have? Is it segmented at all? It's going to tell you how many appendages do you have? What's an appendage? Yeah, arms and legs. Okay, fins, wings, flippers. Those are all appendages, things that stick off, right? So how many do you have and where are they? Okay, those are all questions that are determined by different types of Hox regulatory genes. Head, tail, directionality. Which end is which? That is a Hox gene task. Okay, so really, really important in that body plan that we keep talking about. They are homologous across animals. What does homology mean? When I use that term, you guys, we talked about it in the very first chapter. In what context or what sense did we use the term homology?
It's a trait that's derived from ancestry.
Yeah, it's an ancestral trait, exactly. It's shared in a common ancestor. So Hox genes are homologous across animals. Genetic sequences of those genes, so the nucleotide sequence, and the location on chromosomes, where you find them in different animals' genomes, is very similar. If you look across all animals, you can find the same genes because of that shared common ancestry. So Hox genes are homologous across animals. Here's the other sort of tricky part with Hox genes. They have undergone at least 2 major duplication events. Have you guys ever heard of gene duplication events? Maybe in Bio 1 or other classes that you might have taken. So this happens sometimes. It happens with relative frequency, more than you would think, where a portion of the genome during replication is doubled. Sometimes it's the entire genome, you guys, and sometimes that entire genome is retained, okay? Other times it's a single gene that basically gets duplicated. It's essentially replicated twice. So during DNA replication, which you're doing right before cell division, yes? You're just making a copy of all of your genes so that your 2 daughter cells that occur after mitosis have each an identical copy, right? You guys are comfortable with mitosis, cellular, nuclear cell division. So sometimes mistakes are made. That's where mutations can arise. Well, a duplication is a type of mutation, so to speak. So you can make a copy of just a little portion. It's like double copying. It's over replicating, and then just gets kind of glued in there. So in these cases of these duplication events, you end up with multiple copies of the same gene. And this has happened at least twice to this family of Hox genes. So as animals evolved from the simplest ancestor, something that would have given rise to the sponges, right, and other early lineages, as we end up with more copies of these Hox genes, those genes are free to take on new functions. Okay, so we're going to watch the video, and we'll come back to a discussion about what happens when genes are duplicated to their function, okay, and how that duplication and those new functions can contribute to complexity. So when you're talking about the evolution of complexity in animals, older lineages, more basal lineages like sponges, how do they compare to something like a primate, which is a younger lineage, which includes us, right? Complexity. You or a sponge, more complex. Hopefully, right? We're much more complex than a sponge. Tissue level, cellular level, intelligence, right? Body systems, organ systems, all those types of things. So we're talking about the increasing complexity in animals. And part of the explanation for the ability of that to occur comes from these gene duplication events. When you have a gene duplication event that leads to another copy of the same gene, that new set of genes, they're called paralogs, okay? So a homologous gene exists in different groups because they both have a shared ancestor that gave them that gene. A paralog is a set of genes that have evolved through duplication events, and they may have similar functions or not. They probably don't have identical functions. They may have no function at all, but we're going to talk about that on this next slide, okay? Fates of duplicated genes, but we're going to get to that after we watch this video. It's about 10 minutes. It's a crash course, which usually I make you guys watch on your own, but this one I want to watch together. I think it's going to help us in our discussion. All right, hang on. Let me get a PowerPoint here.
Little kids say they want to grow up to be a scientist. Who do they actually be? They want to blow things up. They want to get picked by a radioactive monkey, which will turn them into a terrifying humanoid battle monkey. Or they want to make a fly with eyeballs on its butt, or like a chicken with fangs. Most of the time, scientists don't get to do that stuff. Like, you may blow something up, but it's either going to be in a really controlled setting, or it will be an accident, in which case it's bad. Like a lab where I first worked, the first lab I ever worked in, I had a blood stain on the ceiling. But if you're a scientist specializing in the amazing new discipline of evolutionary biology, you may just get to make a fly with eyeballs on its butt, or even a chicken with teeth, but no battle monkeys. So evolutionary developmental biology, or EVO for all of us school kids, is new science that looks deep into our genes to figure out how exactly they give instructions to make different parts of our bodies. And as the name suggests, giving us some hot leads into the nature of and mechanisms behind evolution. One of the big things showing us is that animals, all animals, animals are way more similar than we ever imagined. You know how I always hear about how humans and chimps are 98.6% genetically similar. It kind of makes sense, right? Chimps and humans, you can see them kind of look alike. Like if you walk to a coffee shop and there's a chip sitting in a chair and it's like maybe wearing a fedora or something, you might briefly mistake that chimp for a human. You might not even notice it's sitting there. It can happen. But what about a mouse? You are not going to mistake a mouse for a person. How genetically similar do you think we are? How about 85% similar? No, I won't shut up. Humans and mice are 85% genetically identical. So why are mice like little and skittery, covered in white fur, and have beady little eyes, while I can like walk upright in a non-skittery way and have beautiful, deep, mysterious eyes? I'll give you the long answer in a minute, but for now, short answer is it's all because of the incredibly weird and amazingly powerful genes called developmental regulatory genes. Sometimes when we're thinking of genes, we think of the things that code for some useful enzyme, protein, like what is determine what our ankles are going to look like. But those ankle genes don't just come on and off at random. They have to be turned on and off. That's what these developmental regulatory genes do. They activate genes that put the body parts together. They don't tell them how to do it, mind you. They just tell them when or if it's time to get to work. And since they're the ones pretty commonplace, regulatory genes start working rather early in embryonic development. For instance, a kind of regulatory gene called GAP genes are responsible for telling the blastula, that's little hollow ball of cells that forms during the early stages of development, like make a mouth here, let's put an anus over on this other end. Probably the most amazing kind of regulatory genes are the homeobox genes or HOX genes, which kick into gear after the embryo is more developed. HOX genes literally control the identity of body parts, setting up how an animal's body is organized. Like, here's where you put the leg. Here's where you put the tail. And like I said, these HOX genes don't give instructions for how to create legs and tails. There are a bunch of other genes that are in charge of the actual craftsmanship of the body parts. You can think about HOX genes just like a head architects in the construction of the building. They've got the master plan, but they don't do any of the construction themselves. That's way beneath them. Because of this top tier regulatory genes, there are stabs of other genes that act as like subcontractors. Like if a HOX gene tells us to direct subordinates, make an eye here. The subordinates then turn around, activate other regulatory genes to get more specific instructions like, this is where we got to put the collagen, the outer shell of the eyeball, and make some nerve tissue for a retina right here. Again, these second tier genes, and third tier, fourth tier, and on down the line don't actually do anything for work. They just send instructions down the chain of command, adding more specific information to the instructions as they go. It's a really rigid hierarchy. No gene in your body, aside from that very first one, does anything until it's told when and how much to do. So because I know that you're such a sort of intelligent, curious student, I know what you're wondering right now. What activates that first regulatory gene? And how in the name of Bill McGinnis did they tell each other to do stuff? Well, since ego is a relatively new discipline, we don't really know all the stuff that I wish we knew. That's for you to figure out when you become a biologist. Scientists are starting to think that a lot of the human genome that has until recently been considered junk DNA, because it apparently doesn't code for anything, might actually be regulatory genes. For instance, just in the past few years, we've learned that humans have about 230 separate HOX genes in our genome. And they appear on every one of our chromosomes, even the sex chromosome. How regulatory genes are inherited is also still being studied. From what scientists have been able to do so far, most regulatory genes are inherited very much the same way as all the other genes. But for some really early stage regulatory genes, the proteins that they're coded to produce, called gene products, have already been made and are sitting in the egg before it's fertilized, waiting to tell the embryonic cells what to do to get the ball rolling. Another thing that your mom did for you that you probably never thanked her for. So here's a really cool thing. Even though most regulatory genes are inherited, each individual within a species tends to have the exact same DNA sequence in those genes. There aren't even different alleles. So when you think about it, they kind of have to be the same, since all individuals in the species should be built from the same basic blueprint. Like you don't want people walking around with thumbs sticking out of their heads. Wow, this gets me back to me and my beady-eyed friend mouse. HOX genes and other regulatory genes that are the very highest tier, the ones that say like head, ear, eye, ear, not only tend to be the same within the species, they're also very similar across different animal groups, like between all mammals or even all vertebrates. The differences between my regulatory genes and mouse's regulatory genes are way down the chain of commands, where the instructions are the most specific. But the big picture stuff, like you're a vertebrate, you have 4 limbs, and you have hair, and breast tissue, and ear bones, and all that stuff that all mammals have, all those general instructions are the same. And that's why 85% of human genetic makeup is the same mice. Mices. Mouse, mice, mices. Okay, you're very patient, my students. So I've got a surprise for you. We're gonna make some bun eyeballs. In 1995, in a very cool and also totally messed up experiment, a team of researchers in Switzerland took a Hox gene from a mouse embryo, one that said eye goes here, and inserted it into the DNA of the developing fruit fly embryo. But they activated the mouse eyeball gene in a region of the fly that would become the fly's back leg. And so what do you think? I'm not gonna tell you yet. So I want you to guess. Wrong. The fruit fly did not grow a mouse eyeball next to its back leg. It grew a fruit fly eye next to its back leg. Remember, the gene didn't say how to make an eye. It just gave the instruction to make an eye. If it said how to make the eye, you get a mouse eye on the fruit fly spot. Instead, it told the fruit fly cells, make an eye here. And those fruit fly cells had their own instructions regulated by another whole set of regulatory genes. Once they got the order to make the eye, they made it the only way they knew how. That is pretty freaking messed up, but also freaking awesome. Now, in addition to getting me in touch with my inner mentally unstable child scientist, this kind of experiment where you really go back begun to really revolutionize our understanding of evolution. Because we've known that evolution can take place over a really long time, but we haven't really been able to figure out how it sometimes happens really fast. Traditionally, one of the main ways that scientists have explained evolution is through genetic mutations. But an organism would have to do a lot of mutating to evolve from, say, a dinosaur into a bird. It used to be thought that a 50% change in form would require a 50% mutation in genes, which would take a long time, way longer than the pace at which we see things actually evolve. But it turns out that a small change in a regulatory gene up at the top of the chain of command can have huge effects on how birds can actually assemble. To understand how this works, let's look at why birds don't have teeth. So birds evolved from theropod dinosaurs, which are these freaking sweet dinosaurs like velociraptors, which look a lot like birds, but way more awesome and with big razor-sharp teeth. But you may have noticed that birds don't have razor-sharp teeth; they have beaks. Under the old way of thinking about evolution, the loss of the teeth would have had to happen very slowly as the genes make enamel and dented gradually mutated to make less and less and less of each of those things in the living up. And for a long time, that's just how we thought dinosaurs evolved into birds. But there was one problem. It would have taken way longer for all of those mutations to occur than it actually took for dinosaurs to evolve into birds based on the fossil record. Fortunately, EvoDevo is offering us an explanation. A single mutation in the regulatory genes could have shut off the enamel and dentin production, and another mutation in another regulatory gene could have upped the keratin production from the level of mixed scales to the level of make a beak. So birds actually do still have genes for teeth from their dinosaurian ancestors. They're just not expressed because the regulators don't turn them off. But how do we know that? Well, in 2006, a biologist at the University of Wisconsin named John Fallon, who studied bird defects, was looking at some chicken embryos and noticed that they had formed little teeth, like little baby reptile teeth. It turns out that the mutations affected the chicken's gene regulation, allowing the teeth, which were lost to birds around 60 million years ago, to just pop back up again. The same sort of crazy throwback features have been observed in snakes born with legs like their ancestors once had, or blind cave fish suddenly born with eyes. If you turn those genes back on, those ancient repressed features come back. It's crazy. I know. That's so cool. It's all fairly new science, so this is still like in my head, this is like really fascinating. That's a word I made up. Thank you for watching this episode of Crash Course Biology. I hope that I blew your mind, or that you learned something, or that you do well on your test, or whatever you came to watch this episode. If you go already, you can click to catch up on things that you may have missed, or just rewatch the whole episode. It's got it sized in your mind. Otherwise, you don't remember these things. Thanks to everybody who helped put this episode together. If you have any questions,
You don't need the credits. Okay. So does that shed a little bit more light on Hox genes and how they work for you? Those examples are kind of cool, I think. Let me go back to the slides so we can sort of talk about what I wanted to touch on here. So I really like, I don't want to do that yet. I want to go back to part of the video that I really like, that is a great visual representation of what we're talking about here with these master genes I wrote down. Yeah, right here. This is the perfect way to explain it, right? So your Hox genes would be up here. And you turn these on and these turn these on or off, right? And then these are gonna further control the next genes in line. So it's like a hierarchy, right? With the Hox genes in the top being in charge of turning stuff on and off, and then the same thing sort of trickling down. So when we talk about the genetic similarities, so he mentions humans and mice are 85% genetically similar. At which one of these tiers would you imagine we are the most similar to mice? Top 2, 3, or 4? Top, who said that? Why top?
Since that's the genes that in that version.
Yeah, that would be where those are the regulatory genes. Those would be the Hox genes. And he even mentioned in the video that if you look at people, especially, the sequence of those genes isn't even different. Like there are no alleles, there are no versions of those genes, they're just genes. Because you can't change it. The selection on those regulatory genes, especially top tier like Hox genes, is so tight, you can't make a mistake. Because if you do, the downstream effects of it are devastating. Like organismal level devastating, right? Or chickens with teeth, or dinosaurs without, right? So major changes. In most cases, probably not great. But occasionally, like with the chicken teeth thing, you know, something works well. Do you see the point? So the selection on that, so if I have an embryo, and that embryo has a mutation that causes a top tier regulatory gene to malfunction, and it doesn't make, I don't know, it makes the head on the wrong end, that's probably not a viable embryo. You guys with me? The rest of the pathways that are going to happen downstream are going to be all kinds of messed up. So the selection to keep that sequence, those gene sequences, the same is so super tight. Does that make sense? So that's why you see that homology. Now, he also mentioned that we're 98.6% similar to other primates, like chimps. Our closest relatives, chimps and bonobos. 98.6% similar, you guys. Where are the differences that make a chimpanzee and a Homo sapien two different species? Top tier or down here? What do you think? Down here. These are the genes that are super specific, right? This is where you see that variation. And you'll see some here and you'll see some here. But the point of it is that most of that loose ability to change and evolve and create variation through mutations and other mechanisms is going to be happening down here. Because if you get mutations in these higher tier regulatory genes, you have more likeliness of detrimental effects. Yeah? So when we talk about people, do you know how genetically similar you are to other Homo sapiens? How closely genetically related are we to one another? It's like 99.99%. Did you know that? Do you think you're not related to each other because you don't have the same like cousins? But you are 99.99% genetically identical. The 0.01% of our genome that's different, where those different alleles come up, it's all down here, right? You guys with me? So the things that make you taller or shorter, or your hair a different color, or your eyes a different color, or your toenails thicker or thinner, right? All the variations that we see are that tiny, tiny little bit of the genome. The rest of it is tightly selected to stay the same, or at least very, very similar. You guys good? Does that make sense? Okay, that's the sort of takeaway message. Oh, the other thing that he mentioned that I think is really interesting is junk DNA. Have you guys heard that term before? Some of you are nodding. I feel like it's sort of falling out of favor a little bit because we're learning what it does. So I don't know, 20 or 25 years ago, we just thought that there was a whole bunch of code in a whole bunch of nucleotide sequence that didn't do anything. So they call it junk. That doesn't really make a ton of sense if you think about it. So there's so much research going on into what these sequences are actually doing. And as you mentioned in the video, a lot of it is regulatory. So we think we're so smart. We're like, oh, it's all just junk. It's not, right? It's doing stuff. Even if it's not directly coding for proteins, it's controlling the genes that do code for proteins in many cases. So if you're interested in a career in genetic research, there are plenty of questions that are still waiting for answers. That's the best part about science, yes? What we don't know yet. Okay, So let's actually back up just a smidge, back to this slide, and we'll talk about the paralog thing again, and then we'll talk about how duplication of genes that lead to paralogs or paralogous genes, those mean the same thing, okay? How that can lead to the evolution of complexity or more complex features in organisms. All right, so we're gonna do a thought experiment here. You've got a gene, and the genes, this sequence has 4 different functions, okay? And then through some sort of duplication event, whether it's a whole genome duplication event or just a single gene or whatever, this gene gets duplicated. So now we've gone from one copy that can do these 4 different things. Let's say it codes for 4 proteins or unique proteins, all right? Now you have 2 copies of that gene and they're both capable of coding for those same 4 proteins. Now you have a double dose. You guys following me so far, right? So you have a couple of different things that can happen. You can keep both, in which case you are likely just making twice as much of each of those 4 proteins. That happens sometimes, okay? You can get benefits from that. You can get drawbacks from that. Okay? You're just making more of it. But it's more likely due to the fact that natural selection is a thing, and due to the fact that selective pressure will relax if you don't need to conserve the sequences tightly, you see variation more likely than you would see for 2 duplicated genes to just stay exactly the same. So a couple of things that can happen. Let's start with neo-functionalization first, because I think it's kind of the most straightforward. So what happens here is you still have these 2 copies that have been duplicated. The original copy, or one of the copies, stays the same. It retains the same functions. It's still making these 4 proteins. But now this second copy is free to mutate. Okay, hang in there with me. If you are still doing all the things you were doing before, there's no reason you have to do it twice. Okay, so this copy is potentially less tightly regulated by selection to stay the same. Is that sinking in? Okay, so if this copy starts doing something else, if it mutates, it's okay, right? Why? You got to back up. So this opens up the possibility for this second duplicate to do different functions or new functions, neo-functionalization. Okay, so you can get mutations that make new proteins. Okay, because you've got the old copy still doing the job. You're not losing any effects. You're not losing any function. You're just gaining new functions in that second copy. Does that make sense? Okay, that's neo-functionalization. Sub-functionalization is where the first copy stops doing something and the second copy picks up the slack. All right, so that's a possibility as well. The benefit that you could receive from this could also be a dosage thing, right? So now this copy is free to continually specialize on just these 2 proteins. Maybe it gets better at it. Maybe it can make more. Who knows, right? The possibilities are endless. But that's sub-functionalization. So the 2 new copies then split the job. See how that's different from neo-functionalization. So far, so good, yes? And then the degeneration or gene loss is probably the easiest one of all to understand. You just get rid of the duplicate. Okay, so if you have 2 copies and you don't need that second one, there is no selection on the genome to keep that second copy, and sometimes it's just tossed. So organisms do this all the time. If you have duplicate genes or redundant genes or genes you don't need anymore, just chuck them. Because it's expensive, metabolically, energetically expensive to make nucleotides into DNA that you don't need. Okay, so the selection is actually to get rid of it if you don't need it. So these are just some examples of the fates of duplicated genes. All right, now let's go back to talking about complexity. So if you're looking at something like a fruit fly, or Drosophila up here, our model organism, you've got these Hox genes. Compared to mouse and human, we have all these other paralogous copies of these same Hox genes. So different versions of these same genes, now they all have different abilities to sort of specialize. So you can start doing more things. Okay, you can do things like vertebrae, right? Large brain, things of that nature. Does that make sense? So the more sort of flexibility in your genome, the more complexity can arise. You guys okay? So this is a little bit more complex topic than some. Okay, this goes well beyond memorization to like solid understanding of what's going on here. This is another video. I'm not going to play it for you guys, but you feel free to watch it on your own time. It gives a couple of examples of traits in living organisms that are the result of gene duplication events. It's a pretty good explanation of how that can happen, okay, and sort of what it can lead to. So maybe take a look at that if you have an opportunity. You guys good? All right, so for the exam, this video, since we watched it in class as part of lecture, you will definitely see questions that have to do with the topics in that video, okay? For this one, that's an Easter egg, that's a bonus, okay? But take a look at it if you have an opportunity or if you're interested. Questions about Hox genes or duplication events or chicken teeth? Everything good? All right. Interesting? I'll take the nod. I think it's cool, right? It's kind of weird and interesting, which is the best stuff. All right, the rest of the chapter is going to talk about classification features. So these are traditional ways that we classify animals into taxonomic groups. Okay, we're not yet talking about molecular systematics as classification methods. Okay, we're just talking about traditional ways to classify animals into groups. Now, interestingly, when we do look at the molecular systematics, most of the relationships that are determined based on these traditional classification features hold up. So the DNA says the same thing as the morphological anatomical classification features that we've been using for hundreds of years, which is pretty cool. Why do you think that is? Well, these morphological features are dictated by what? Where does your body plan come from? Where is that predetermination coded? DNA, right? So we do see lots of examples in lots of different organisms of convergent evolution where like they look the same, but it's not a DNA thing, it's an environmental thing. But in a lot of these cases, these are really strong lines of evidence for relatedness because they do hold up to that molecular scrutiny, which is kind of cool. And we'll talk about some examples of when that works and others when it's still in question as we go forward. All right, the first feature is symmetry. What does it mean to be symmetrical? Even on both sides. So if you have a single plane of symmetry, then you have two equal sides. That is called bilateral symmetry, like our friend the goat or yourself. Yes, you have a single plane of symmetry, one line that you can cut yourself in half and you become two relatively equal parts. Right? We're not perfectly symmetrical, right? Your heart's a little off center, that kind of stuff, right? Appendix is on one side, your liver is further over than to one side than the other, but largely we're symmetrical, okay? That's bilateral symmetry. Radial symmetry, think about like a circle having a radius, right? What's the radius of a circle? Half the diameter, yep. So it's when you draw a circle, put a dot in the middle and go to the side, okay? I'm gonna move the board so that you can see. I don't know if you guys can see what I'm drawing. Let me erase it. All right, hopefully you can see and zoom. All right, here's the circle, here's the center, there's the radius, yes? So if you start drawing radii out from the center, that reminds me of sort of spokes on a wheel, doesn't it, you guys? Or like a pie or an orange that you slice, okay? So radial symmetry means that you have multiple planes of symmetry. Here's one, here's one, here's one. You can cut that organism in any of those planes and still have relatively equal halves, okay? You can't really do that with an animal that displays bilateral symmetry. There's only one way to cut it in half and get two equal parts. Radial symmetry, you have multiple planes of symmetry. See the difference? So something like a hydra, radially symmetric, okay? No right and left, no front and back, just same all around, okay? And then you have asymmetry, which means what? What do you think? Not symmetrical at all, okay? Guess who this is? Those weirdos, the sponges. Okay, parazoans don't necessarily display any type of symmetry at all. So that's one way to classify animals. We'll look at where these traits fall on the phylogenetic tree here when we get through the rest of it. All right, the next way to classify is through number of germ-line tissues. So here we are. I told you guys 2 lectures ago that we would eventually stumble upon a schematic that would show you those endoderm, ectoderm, and mesoderm germ-line tissues, and that's this slide now finally. So this is what we've been talking about. During development, early tissues form in germ layers. Radially symmetric animals have 2 germ layers. They only have endoderm and ectoderm. Okay, so that's the picture on this side. The outer blue ring is your ectoderm. The green inner ring is your endoderm, and then you've got some brown substance, some sort of non-living matrix in between, some sort of something that separates the 2, but no mesoderm. Okay, so if you are radially symmetric, like a jelly or a hydra, you also only have endoderm and ectoderm, just the 2 germ-line tissues. These types of animals are referred to as diploblasts. The term blast, if you are going into any kind of medicine, you will see this term over and over and over again. It means precursor, like comes before, or turns into something, or gives rise to something. Okay, so diploblast basically means you have 2 layers that become stuff. Diplo meaning 2, right? And then blast meaning precursor. So you've got 2 germ-line tissue layers. Bilaterally symmetric animals have 3 germ layers: endoderm, mesoderm, and ectoderm. And these guys are referred to as, guess what? Triploblasts. 3 things that become something else. 3 precursor layers. Okay? And as we've discussed before, each one of these germ layers gives rise to specific sets of tissues and organs. So here's your schematic of a triploblast with ectoderm, mesoderm, and endoderm. Okay? Interestingly, the same place on the tree where bilateral symmetry comes in, that 3rd germ layer also comes in. Okay, so bilaterally symmetrical triploblasts, radially symmetric diploblasts. They go together. Okay, I'll show you a tree in a minute. Okay, the 3rd of 4 classification features that we'll talk about is the presence or absence of a body cavity. When we're talking about a body cavity, we're talking about internal space for structures. Okay, triploblasts are the only ones that might do this. So if you're a radially symmetric diploblast, you do not have a body cavity at all. And the term that we're using to describe this cavity is called a coelom. Looks like it says solum, but it's coelom. Okay, the coelom lies between the digestive system and the body wall. What does that mean exactly? Let's look at these 3 examples of these 3 different sort of classifications, and we'll sort of describe it as we go forward. If you are an acoelomate, you have no body cavity. So you have endoderm tissue on the inside. Here is your digestive tract, the tube that formed down the middle. So remember the ball, remember the blastula, that then becomes the gastrula. So this is like a cross section of that sphere. You guys tell what you're looking at? Like you took the gastrula and you chopped it in half, and now you're looking into it. So you've got the empty tube or the hollow tube of the digestive tract in the middle, surrounded by endoderm, mesoderm just outside of that, ectoderm just outside of that. All of those tissues that form from those germ layers touch each other. There's no space. That is an acoelomate. A meaning absence of, coelom meaning body cavity. Okay? So things like flatworms, which we'll look at here in just a few minutes. You can also be a eucoelomate. What does EU mean? Like eukaryote or eudicot? True. Yep. So you see it all the time, right? So eucoelomate means you have a true coelom or a true body cavity. What makes a body cavity true, okay, means that it is fully derived from mesoderm tissue. Okay? So look at this. This is the same sort of schematic, but in a eucoelomate. You've got ectoderm externally, mesoderm inside of that. And you've got this coelom, these spaces that are entirely embedded within mesoderm tissue. Okay? Inside of that, you've got endoderm, and then you've got your digestive tube. So what we're talking about here is that true coelom. Let's back up to this bullet where it says, true coelom arises from mesoderm only and is surrounded by mesoderm-derived tissue. Well, what comes from mesoderm? Connective tissue, okay? Muscle tissue. The coelom, the true coelom, houses things in us, like kidney, spleen, and major vessels of the circulatory system. So I'm not sure how familiar all of you guys are with your own internal anatomy, but what you're basically looking at here, ectoderm tissue is going to give rise to things like skin and nerves. Okay, so this is like, you think of this as like your skin. Under your skin, you've got connective tissue, bones, cartilage, you've got muscle tissue, right? You have an empty space inside of your abdomen that holds your organs that are not your digestive tube. Okay, so your digestive tube goes straight down the middle, from mouth to anus, open, right? Food goes in, travels through, digested, nutrients are extracted, food goes out. Yes, so that's just a tube. Lining that tube is tissue that is derived from endoderm, epithelial tissue, linings and coverings. Okay, remember that. So this is lining your digestive tract. But outside of that, you have a muscular body wall. Yes? That's holding your internal organs. Things like your kidneys, things like your spleen, things like your major circulatory vessels. They're not part of your digestive tract, but they're not also embedded in your body wall. They're in between. You guys all cool with that? That's what a coelom is. So your cavity that holds all of your internal organs that aren't attached to your tube going down the middle, that's all in your coelom. Okay, so you're a eucoelomate. There's a third option. You see it in groups like roundworms. So these are not that common, but you can see a pseudocoelomate, and that means they have a cavity, but instead of being fully embedded in mesoderm-derived tissue, it is endoderm on one wall, the inner wall of the coelom, and mesoderm on the outer wall. Okay. I'm not going to ask you guys to get into tremendous amounts of detail about these. Okay, or even necessarily recognize which group is which. I just want you to know what a coelom is, okay, and that you can have a true coelom, fully mesoderm derived, a false coelom or a pseudocoelomate, right, would have a false coelom, meaning it's a space, but it's not fully embedded in mesoderm, or you can have no cavity at all, okay? Just the classification feature, if you see a question about it, it'll be like one question on the test. So don't spend your whole life worrying about coelom. Okay, just the feature. Sounds good? Does that make sense? Enough to move on if there's only going to be one question on the test about it? Okay, that's super important, my humble opinion. All right, and then the last of the four of four, is whether you are a protostome or a deuterostome. Okay, so this is, as promised, the discussion on which direction your digestive tract forms. So if you are a eucoelomate, you have a true coelom and a true digestive system with a cavity surrounding it. You can be further divided into groups based on which opening of the digestive system forms first during embryonic development. Either the blastopore becomes the mouth and the other end keeps forming, that tube keeps forming, and the other end becomes the anus, or vice versa. So the blastopore might become the anus, and then the tube continues to form through, and at the other end, that's mouth. So if your mouth forms first, you are a protostome. Stome, think like stomate in a plant. It's an opening, right? So stoma is mouth. And proto is first, like a prototype. If you invent something, your prototype is the first one you make, right? So proto means first. So first mouth, protostome. Deutero means second. Deuterostome, second mouth. This seems like a really arbitrary reason to classify an animal, doesn't it? Seems like who cares? But interestingly, this will also, the relationships that you can determine based on protostome versus deuterostome development also hold up if you look at the DNA. So it works. It's weird, but it works. All vertebrates, including us, are deuterostomes. And the only other group of animals that are also deuterostomes are echinoderms, which are sea stars, sea cucumbers, sand dollars. That group, that phylum, Echinodermata, are the only other deuterostomes. They're our closest relatives in the animal kingdom. Did you know that? You are more closely related to a sea star than you are to a worm or a butterfly. Pretty cool. I don't know. I think so. Anyway, that's that. Okay, so those are your four classification features. Just to recap. Symmetry. Germline tissues. Do you have two or do you have three? Do you have a coelom or not, or a fake one, a pseudocoelom? And then which end of your digestive system forms first? Okay, those are your 4 classification features. Let's look at them on the tree and sort of pull the whole thing together here. All right. Here are the metazoans. What are metazoa? Who's this? All what? Animals, yeah, so metazoans. Parazoans over here. Yes? This is the first node that divides into different groups. Okay, so metazoa is a monophyletic group containing everyone. If you cut out the organisms that have no true tissues, you remove the parazoans, now you're down to the eumetazoa. Okay, so tissues is the first feature, the first node, the first feature that divides animals at that node. Okay, so that hopefully makes sense. When you get to the next node on the tree, you're going to divide animals again into bilaterians versus radially symmetric. Radiata versus bilateria. Okay, radially symmetric versus bilaterally symmetric. Okay, so that's at that next node. The only radially symmetric groups that are shown on this tree are the cnidarians and the comb jellies. We'll look at pictures of these guys in the next chapter when we get into the group, but the cnidarians include your jellies. Okay, jellies, anemones, corals, things like that. And then comb jellies kind of look like jellies too. Well, those are ctenophores. We'll take a look at those too. So those are radially symmetric, but look here, they're also diploblastic. What does it mean to be a diploblast? How many germ-line tissue layers do you have? 2. So radial symmetry and diploblastic are at the same division. Okay, and then look up here. Bilateral symmetry goes along with how many germ layers? 3, triploblasts. So both of those features pop in at that same node, okay, that division. The next node here separates you into protostomes versus deuterostomes. So here we are, we're in the chordate phylum. So we are chordates, vertebrates are all within this group. And then the starfish group, the echinoderms. We're the only deuterostomes. And then the protostomes include everybody else, arthropods, which are things like insects, crabs, spiders, things of that nature, and roundworms. And then flatworms, rotifers, annelids like earthworms, mollusks like octopus, they're all up here in protostomes. Okay? So you can sort of see where those classification features land on the phylogeny. Right? So that helps a little bit, hopefully. The next node are dividing the protostomes into ecdysozoans and lophotrochozoans. I'm not likely to ask you guys too much about that, if anything, because we're not really going to go into it in our diversity chapters, talking about the processes at all, so I wouldn't worry too much about it. But since it's on a tree, I'll quickly mention what it is. Do you guys know what ecdysis is? You heard that term before? It's another term for molting. You know what molting means? It's shedding your skin. Yeah, so if you have an exoskeleton, not really your skin, your exoskeleton. If you have an exoskeleton, what does that mean? If you possess an exoskeleton, hard parts are where? On the outside. What do you guys have? An endoskeleton. Your hard parts are on the inside and the fleshy stuff is wrapped around, right? So if you have an exoskeleton, the only way to grow bigger is to top that exoskeleton off and build a new one underneath. Yes? So you have to get off the exoskeleton first and sort of puff up and harden again. Okay, so that's ecdysis. So that's the group that does that. Arthropods and roundworms, they do that molting process. And then lophotrochozoans don't. But the lophotrochozoa is named for their feeding structures and their larval features, which we're not getting into, so don't worry about it. But it's kind of interesting just to see that these fall out into another category that molts. Anyway, that's more of a note than, I don't even think it's understood that. So don't sweat ecdysozoan versus lophotrochozoan too hard, okay? All right, the animal phylogeny has historically been determined by this morphological data. Symmetry, protostome versus deuterostome, et cetera, and the fossil record, which is also morphological, right? We're looking at shape, we're looking at feature, we're looking at characteristics, and that's how we built this animal tree. But then, check it out, with the advent of systematics, and looking at the DNA, many changes have been made, but they're mostly within these groups, okay, moving organisms around. But this is largely held up by the systematic data, okay? So deuterostomes are still deuterostomes, okay? And it all sort of falls out, even with looking at the DNA. So the DNA supports most of those relationships, okay? even though things are gonna move around. So we'll talk about some examples of that later. All right, this is a quick quiz. No points, just try it. Read the tree. Which of the following statements is false? Based on this tree. There's a couple of groups on here that we haven't talked about yet, that you guys can read if I was genetic tree. I'm certain of it. Eumetazoans have specialized tissues and parazoans don't. Is that true or false? True. How can you tell? By the tree.
First, branch.
Here, right? Yeah, eumetazoans, specialized tissues, parazoans, no tissues. Looking at the tree, right? Lophotrochozoa and Ecdysozoa are both bilateria. True or false? And did you say something? True? Why do you say true?
Because looking at it, branch also both go into the bilateral.
Here, can you point at it? Push the button, point to it.
So this bilateral is split off here.
Yep,
So we got those whatever.
yep. Yeah, you guys all see what she's talking about there. They're both over here. They follow back to this node where you see bilateria come in. Good. What about Acela and Nidaria? Both possess radial symmetry. Can you say something? I just heard sound. I think it was you again, but I'm not sure.
I think it's false. I'm just really checking.
Okay, she says she thinks it's false. Anybody else want to back her up or contradict? Jacob, you say false. Why do you say false?
Area is only regional because it's splits off at the second.
Oh, that's what you're talking about?
Splits off down here.
Yep.
Where Aciola is bilateral because it's
Yeah, it's later on the tree. Then that knows where bilateria comes in. Good. So that's our false one. So we already got our answer. But let's look at the last one just to practice. Arthropods are more closely related to nematodes than they are to annelids. Well, it must be true, right? Since we already identified C as false. But how do we know that? Where are arthropods? Up here, right? And then nematodes right here. And then annelids are down here. So do you think that nematodes and arthropods are more closely related to each other than annelids are to either one of them? Yeah, right? How do you know? Proximity, right, on the tree, but they share that node right there. So those groups would be sister on this tree, and annelids would be over here. So they're not distantly related. I mean, depends on what you're looking at. We're looking at the entirety of animal phylogeny, so distance versus closeness is all relative. But they're here, they're also protostomes, so they're in that group, but these guys are definitely more closely related. Good tree reading exercise? Yeah, cool. So you might see a question like that, where you don't necessarily have to have this knowledge. I told you I wouldn't ask you about Lophotrochozoa and Ecdysozoa specifically, but I can ask you about reading it on a tree, right? See the difference? Okay. All right, cool. And then another video. This one is the same crash course guy, Hank again, probably about 10 minutes. I'm not going to play it in class, so again, it's an Easter egg for bonus questions, but it's a really nice summary of these developmental features, these classification features of animals. And that's the end of 27. But I think we're going to keep going because we've got like almost 15 minutes. So I want to introduce invertebrates today. You guys okay if I do that? Since we dipped out early on Wednesday last week. That's the tough day. Power through till 12:15. 13 minutes. You guys can do it? All right. You can do it, I can do it. You know I can do it. I'll just stand up here and talk to you about this weird stuff all day. All right, invertebrates. What a lovely picture of an echinoderm. Yes? Okay. Invertebrates are the majority of animals. Okay? 97% of all known species of animals are invertebrates. Did you guys know that? What is an invertebrate? What makes an invertebrate an invertebrate? Well, it's an animal, right? What does it lack? Backbone, no vertebral column. Now, here's a question. Is the backbone a bone or is it a collection of bones? Collection of bones, yeah. You've got vertebrae that are stacked. They actually have really tight joints in between each one of them. You've got your discs, your intervertebral discs that we have as upright vertebrates. So in between each of your two, let's say you've got one and then two, right? There's not a lot of movement that happens in between those two, but if you collectively have a whole bunch of them, like 20 or so, you get a lot of flexion. Does that make sense? So it's not a single bone, it's a bunch of stacked bones that can flex. But invertebrates do not have that. They also lack a cranium. Do you guys know what a cranium is? Yeah, it's a brain case. So your skull is your cranium. Okay, so we'll look at some examples of different types of crania that invertebrates don't have it. Most invertebrates also lack an endoskeleton. We just talked about that, right? So if they have a skeleton at all, it's going to be exo. It's going to be an exoskeleton. But I said most. So what do you think that means? Exceptions, always with the exceptions, right? Many species are aquatic, and there are likely, oh, this is going to be a shock, you guys, right? Likely many more species that have not been identified or described. That's the same thing we say about every group, right? There's a bunch. There's probably a lot more we don't know about. All right, so back to this. If 97% of all known animal species are invertebrates, why is there such a vertebrate bias? And when I say vertebrate bias, what I mean is if I walk into a classroom of fifth graders and I say, name an animal, and I go around the room, is any kid going to name something that isn't a vertebrate? Or are they going to say dog, cat, bear, chicken, snake, lizard, frog, cow, right? Every single one of those is a vertebrate. Why do we think that way if there are only 3% of all the known species of animals that are vertebrates? What do you guys think?
That they're most closely known, and also I think vertebrates because that's what they come into contact with.
Yeah, familiarity, right? That's what you are. That's what you come into contact with. Unless you keep like, I don't know, tarantula as a pet, it's possible. We've got 2 hermit crabs at our house, but we're weirdos, so you know how that goes. Yeah, there's a vertebrate bias when you think of animals because you think about yourself and the animals that you're most closely sort of interacting with. So it's kind of interesting to think about the fact there's so much diversity outside of the vertebrate group. All right, here is a list. I know you guys love lists of terms that you've never heard before. So hopefully some of these are not as unfamiliar, or you've heard at least some of these before. We're going to go through each of these groups and we're going to talk about their features and their characteristics in more detail, but this is a nice reference. And here's the tree again. Okay, so this is sort of the summary slide as we get started. We're going to talk about Porifera, Ctenophora, Cnidaria, Platyhelminthes, Neomatoda, Arthropoda, Mollusca, Annelida, Echinodermata, and Chordates. Are you guys ready for this? I am going to ask you to know these words. Okay, so this is a list of terms that you will need to know for these groups. All right, by the time we get through the lecture, you will hopefully be starting to make connections that will help that be more easy to remember. Okay, but I will ask you to identify a group based on things like characteristics or species that you would find in that group. So sort of notable organisms in that group. Okay, so we're going to go through that. Chordates have an asterisk off to the side because they fall in both the invertebrate chapter and the vertebrate chapter. So remember, there's not really a dividing line in between invertebrates and vertebrates. They're all animals, but the way our book does it is it splits it into 2. So you don't have all the groups in one big chapter. All right, so there's an overlap when we get to chordates. All vertebrates are chordates, but there are a couple groups of invertebrate chordates that we'll cover when we get to that point. Okay, so those are your invertebrate phyla, and then you got chordates. which is a largely vertebrate phyla, but a couple of inverts that are tucked in. So that's where we're headed with that. All right, let's see how many groups we can get through in 6 or 7 minutes. Porifera, we have discussed some of these features, most of these features we've already discussed in previous lectures. Who are the poriferans? Sponges, basal group, right? Early branching lineage, no specialized tissues. We talked about that. Some cells can specialize function. They can shift and change function even, and they don't display symmetry necessarily. Although sometimes their larvae can be symmetrical, but as a whole, just sponge, okay? Filter feeders, we talked about choanoflagellates, we did protists. That's the sister taxon that's considered a protist if it's single-celled eukaryote. They look like choanocytes. You guys have seen this picture before when we looked at choanoflagellates. It's back. This is a choanoflagellate. That's a single-celled eukaryotic organism. This is a collar cell or a choanocyte. These cells live inside the gastrovascular cavity of the sponge, and they're involved in filter feeding. Okay, so the interesting thing about this is that they look, morphologically, they look the same. Okay, so this is strong evidence of early animal morphology. Okay, not necessarily that the first animal looked like a choanoflagellate, but that there's deep homology there, okay, between that sister group and the basal group of animals, which is your sponges. Okay, so just some evidence. And again, when you look at the DNA and you look at molecular systematics, there is solid evidence that these are the oldest animal lineage or the most closely related living organism to what the first animals would have looked like. Okay, so not that they were sponges, but that sponges are the most basal animal lineage. Do you guys understand the difference between those two statements? Okay, so it wasn't like we all came from sponges, but that sponges are the earliest branch from the extinct common ancestor of all animals. Okay, so it's a little bit semantics there. With sponges, a feature that you might see sometimes are these structural supports. Sorry, I can't speak. Structural support formations called spicules. Sometimes they're made out of silica, like glass sponges, so those are kind of cool. Or calcium carbonate. You're going to see calcium carbonate throughout the animal groups. Those are basically anything that makes a shell, making it out of calcium carbonate. They pull calcium ions out of the water, carbon dioxide out of the water, building essentially limestone. The whole will become limestone later. So those are your poriferans. I always like to look at the part that says pore. Because what's a pore again? Pores are openings, yeah. And what are sponges full of? Pores, yes. Sponges are porous. We actually use the word porous to describe a sponge. Even like a sponge you think powder with, yes? Porous. Okay, so that's helpful, I think. All right, let's talk about the ctenophores. These are one of my favorite groups as well. If you have ever been to the Tennessee Aquarium, you may have seen these. I can't remember if they have them at the Georgia Aquarium or not. I've only been there once and it's been a while. But these guys are also called cone jellies. So they're not jellyfish. They're in a completely separate group from jellies. Okay, we go back to the tree. Here are your poriferans, basal group. Here are your Nigerians. That's where your jellies live. Here are the ctenophores. They're on their own branch. Okay, they're also radially symmetric and diploblastic, so they share that in common with jellies, but they're their own thing. Even though they kind of look like jellies, they're different. Okay, they're really cool looking though. They have these the way that the light reflects off of their little appendages makes them look like they're sort of almost like neon lights. Really, really cool looking. These guys are a confusing group though. So I put this tree on here. I'm not going to ask you guys to read this tree or talk about this paper at all. And I didn't even put the link to the paper. So you're in luck. It's not even there. But what I'm talking about here is this recent research that's been going on trying to figure out, are these guys the basal group or are sponges the basal group? And I know that sounds like a riveting question that's worth devoting your entire career to, right? But the more we can learn about the groups of animals that we don't know that much about, the more we can learn about all animals, which includes us. Right? So this is an interesting phylogenetic question. They don't have Hox genes. Okay, so they lack that regulatory gene that you see in just about all animals. They also lack genes that support the development of nerves in other animals. So they have some missing links, some missing genetic links that would allow us to make those sorts of classifications. But they do have nerve cells. We're just not sure about the developmental pathway that feeds to them. They also have rudimentary muscle, which sponges do not have. Okay, so this indicates complexity that the sponges lack. Okay, so that's evidence that they are further up the tree, closer to Cnidaria than sponges are, with sponges falling as basal. But there are some other interesting pieces of evidence that say that maybe the ctenophores are basal. So this tree here on the left says, here's your outgroups. These are groups that are not animals. Okay, this would be something like Coena flagellum. Okay, so that's an outgroup for comparison when you're building this tree. Then you've got the first node putting ctenophores basal, and then periphera up here with the other animals. Okay, you see how that works on that tree? Yes? No? Pretty okay with that tree? How is this tree different on the right? This is just more tree reading practice. What does this tree say that's different than this tree? Who's basal on this one? Periphera. See, here's your outgroup, unrelated group. Peripherans here, basal, and then ctenophores up here closer to the other animals. Okay, so these are two competing hypotheses. This comes from this particular paper that I'm referencing here that I didn't give you, so that's good. This paper is actually saying that this model is not as well supported as this model. So with improved analysis, more complex analysis of the molecular data, this hypothesis is better supported. So the answer is still somewhat unknown, but all the most recent evidence supports that indeed sponges are basal. And ctenophores fall in next to cnidarians, like you see on this tree here. Okay. I may ask you about that, just to like which one is which. Like, which of these hypotheses is best supported? Which we are going to stick with sponges. So that was what we talked about. We can change it. That's still best supported. I won't necessarily ask you anything about data heterogeneity or anything crazy like that. But I always like to mention these because the research is ongoing, and that's always cool. I think like our book may be out of date in a year when these new discoveries are made, which I would think is why science is fun, right? All right, 12:15, you did it. Good job. We'll pick up with cnidarians on Wednesday.